So Darwin said in 1871 that little is known about the functions of the brain, but we can perceive that as the intellectual powers become highly developed, the various parts of the brain must be connected by the most intricate channels of intercommunication. And that was prescient because he didn't really know about the nervous system. And I don't believe that Cajal and Darwin ever met, but Javier can tell us maybe. I have not found it. So what I will do today is to introduce here, to say something about why it is necessary to map the cortex. You're doing that in the big project that you're studying all at different levels. And I will use that to show you how connection patterns led us to link them to a universal and classically known principle. And I'll show you why this is important to understand a lot about the brain, and how going to development can help explain patterns. This has pushed us into explaining things in development. It made it necessary to do so. And how fundamental principles can be extended to understand another variation in the brain, the plasticity, stability, continuum of areas, and how all of the above can explain the severity and variability seen in psychiatric disorders, which is what some of you have already been talking about. So, mapping the cortex. To study connections, it is necessary to have a roadmap, an actual map of the cortex. A map is like a compass to an explorer. So you need your compass. So, and Brodmann, this is the map that a lot of people are familiar with. And it was based on the idea that all areas have 6 layers. He made some modifications on that. But basically, Brodmann tried to map the cortex by seeing how there are variations in areas. So for example, you look in the back there, the one here, and how is it different from 17, as it was known then, from V2 and so forth. But to do that, you have to rely on a lot of features that are very frequently subtle. So this is why maps constructed using this principle have been so varied and difficult to do. Earlier than that, people have already proposed that the cortex varies in a systematic way. And what this meant is that the columns, if you take a column across a cortical area, in different areas, they are unequal in depth, in the density of neurons, lamina structure, etc. And this is the opposite of what people have proposed about columns. You've seen one, you've seen them all. This is not true. And so we have seen that systematic variation was discovered independently in different species by different investigators, and they didn't know about each other. There was not a lot of communication, starting from the 30s, 40s, and in the 60s, with Sanidis. And these people worked in different continents. Some were in Australia, some were in South Africa, some were in Europe, and in other places. And so they discovered it almost like a mathematical principle that there is a variation in the cortex. And this seems to, I will emphasize that this is really important to do. So what do we mean by that? So systematic variation says that the cortex is unequal, but it varies in a systematic way. So if we go to the limbic areas, and here you can see on the top is kind of tilted view showing the medial and some of the basal view of the rhesus monkey brain. And there is an inner ring which is black, and there is another ring which is gray. And those are the limbic areas, and they do not have 6 layers. The black area, they only have 3 identifiable layers. the gray have only 4. And there is a differentiation from there, as you see by those arrows, away from it. So basically, if you look at it as a base of a stem, and everything is around it, grows around it, and differentiates from there. So the variation that I will focus on here is an overall structure. How many layers do we identify? The density of neurons by layer. 2 areas can have very different features, but very similar structures. Think of it this way, is if you're putting people in categories by, let's say, a feature like height. So you can put people of the same height in one group and people of a different height in another group. Now we know that the people in group X, even though they have the same height, they look different. I can tell the difference between individuals because of their facial features and so forth. But the facial features is what Brodmann was doing and some other maps. So I will focus on this. So look at it that way, because it is something that is simple, but at the same time elusive. So we can look at this systematic variation. And this is in a cartoon one of my students some time ago made so that people can see it. And basically you can go from these limbic areas, which are either agranular or dysgranular, and you see a differentiation, not only in layer 4, which is granular here, but also in the other layers. And I will show you that this is an important feature. And so what Steve did some time ago, he looked at a lot of features. He looked at the density of the layers, different layers, the different types of inhibitory neurons, and a lot of other markers. And he came up with 17 parameter dimensions. And basically, with my colleague, Klaus Hilgertag, who's a computational scientist, used an NMDS analysis to cluster them. And you can see the limbic areas are clustering over here to the left. And what I will call laminated is the ones that have 6 layers. But there is a differentiation within them as well. And they cluster very nicely. And you show, so basically what these dots show is how close 2 areas are. It means that they are more similar than areas, let's say, that are very far from these regions. So doing another analysis, a cluster analysis, we see the exact same ordering of the position. So there's something really interesting about this difference that we have seen, and you can study it quantitatively. So why is this important? The cortex is laminated, and it is a vast communication network, and it's interconnected by a large set of connections. And the question is, which neurons in which layers are involved in each set of connections? So people who have done work who use neural tracers to study this for the first time found that for every connection that goes in one direction, there is another one going in the opposite direction. And they started in the visual system, and because it's easy, you have a handle on the input, and they found that if you, there is a pathway from the primary area to a secondary area, neurons are originate in layer 3, and they end up around layer 4. And going in the opposite direction, the projection neurons are found in the deep layers, and they terminated broadly in the upper layers. So this was something that people did not know until there were neural traces to do that. And they called what we show here is in red as the feed-forward pathway, because they thought it has something to do with the processing of information and the sequence in the processing. And going the other way, they called it feedback because it was going in the opposite direction. So So the mini synthesis here is that cortical connections are reciprocal, but they are unequal in laminar distribution, as I showed you, and in strength. And so there is reflects the non-uniformity of the cortex. So at about the same time, working on the opposite side of the brain in the prefrontal cortex, and we were using traces for the first time. And before that, I didn't really know much about anatomy. My doctoral work was in physiology, and I wanted to understand the layers. But one of the things that I saw very early on was these areas that we see as limbic, that you see here on the left, they behave like feedback systems, what people were talking in the sensory systems. And so basically, when this cortex projected out, it did so from the deep layers. And if you see on the right side, C and D, when that cortex that has better lamination projects out, most of the neurons are found in layer 3. So it really was not restricted to the sensory systems. We could see this also very far away from the sensory system, all the way in the frontal lobe. So something interesting was happening. So this is the limbic areas that do not have 6 layers. Laminated areas, that's how we'll be referring to them, they have 6 layers. So after a lot of these experiments and asking questions to people who were doing this work in the sensory systems, such as all of the neurons are in layer 3, what do you think? You know, if you go away from it, got some answers and show that it's really not one way or another, all or none. So we link the cortical architecture to connections. And basically, that's what we now call the structural model. And so, which says that the structural relationship between areas underlies the pattern of their interconnections. It's the relationship that is important. So this, it is going, feedback is essentially reflects the pathway that goes from a less to an area with more elaborate structure, as you see there in the blue. And at the bottom, I show you the cartoon of what it would look like. So that's what feedback reflects. And feed forward is in the opposite direction. But the model also predicts what will happen if you have very small differences and it's going from lower to higher or higher to lower, or sometimes you don't see a difference at all. And in that case, more layers are involved, in the cellular layers. So this is what we found earlier. So to show you that this actually works everywhere else, this is another student of mine, Maya Metala, and now she has her own lab, very successful, and looked at the connections between prefrontal areas and the parietal cortex over here and the parietal. So this is opened up to show what is happening. And so she looked at the laminar structure of these parietal areas and using a lot of features of this area, the density of neurons, etc. And then this is how they came up with an MDS analysis. And this is the connections. And I think that it doesn't really take too much of an imagination to see that these two are very highly correlated in the way they are. So it works in a lot of those. So what was happening in the sensory systems, what people call feed forward, really goes from an area like this is a V1 to V2. They both have 6 layers. They're both laminate, but as you can see from here, this has better lamination than V2. They both have 6 layers. So it's really a difference that you can see in that. So here is you've seen this before. This is the primate hierarchy that people use from Helen and Van Essen, and they took a whole lot of connections that people had published and essentially put them in a hierarchy based on the laminar origin and termination. This is not a predictive model. It's a post hoc analysis of a whole bunch of data that were out there. It's very useful because it people can use this database like we also use. Where it says feed forward and feed back, we put these arrows there to show you what the gist of it was. So this is the exact same data set, and now it's been interpreted in the light of the systematic variation, exact same data. So D1 here is on the outer ring, is the most differentiated one. And these are the limbic areas. Hold it around so that you would see what happens. And basically, there is a lot more of these structures that are very highly differentiated. And what this shows, we use the connections, those connections that you saw earlier, to see how areas are linked. And if they were the same type or very close to it, and we did this in black, and then over 2 levels, we did it in blue, and red is 3. And I think that if I ask you which dominates, I think that you would see that black lines dominate. So in other words, the like connects with the like. And it doesn't have to be neighboring. They can be very far away from each other. This also, for example, explains why the frontal eye fields that I showed you earlier and the lateral parietal areas are very strongly connected, even though as a distance, they're very far from each other. But what this shows is they're part of the same ring. So they are equivalent in the height analogy that I was telling you about earlier. So as I just showed you, the model predicts the strength of connections, the likely presence of connections or absence. If somebody says these 2 areas connected, if we've never done this experiment before, you say they're very likely connected based on some of these rules, or they're not likely to be connected. It's not perfect this way, but you can do that. So let me have a short summary. And basically, it's the cortical systematic variation underlies the pattern and strength of connections, and areas that are, have comparable laminar structure are more strongly connected. And the phylogenetically ancient limbic areas have the simplest laminar structure, either 3 or 4 layers that we can identify, and behave like feedback systems in their connections with laminated areas that have 6 layers. But if they talk with each other, they can also be feed forward. So the interesting part about that is that it's all relative. So cortical systematic variation, since it is important for the way the cortex is connected, how does it arise? So we thought, well, all of these patterns may be laid down in development. And let's take a look. What do we know about development? Developmental neuroscientists have told us that in the laminated cerebral cortex, development starts from the bottom. The layer 6 neurons are laid down first. And then it goes to other layers. So it's really something that is counterintuitive. It's sort of like boarding a plane from the back, and the people who sit in the very back sit down there first, and then other passengers come in front of them. Not very efficient from the way we think about it, but that's the way it goes. So we thought there was a hypothesis that we had for development that these limbic areas on the left here stop developing and the laminated areas continue to develop. And that's how you come up with more neurons and higher densities. But we didn't really have the data to know that this is maybe what is happening. So I asked people who were doing developmental work in Rhesus monkey, because all of what we have done was in the Rhesus monkey. And Pashko Rakic was one who did some of that work. And at some point, he published this paper, which has had 3 frontal areas, prefrontal areas. And what you see there in the blue line over here, this is the development of embryonic E40 all the way up to 100 here. And this is the development of the layers. And so the development of this area here is the limbic area, and you see stops developing at E70. Luckily, his other example was an intermediate area. You laminate that intermediate, it was area 11, and these continue to develop until E80, to all the layers. 46, which has a really nice lamination in the prefrontal cortex, continued until E90 here. And this one here, if you cannot read the title, is V1. It's the one that has more neurons in the cortex. And in primates, it's a very important, the visual system is very important. So it's a quasi, some support for the hypothesis that these are late down in development. Do these principles apply to the human cortex? And we heard some wonderful presentations on this, where we cannot study monosynaptic connections as in animal models. And there was somebody working with us in the lab, Miguel Angel Garcia Capezas. He came to me one day, he's a neuropathologist, pathologist, and he said, you know, I have some fetal tissue that he used as a pathologist from accidents of pregnancy, you know, sort of the implantation wasn't right because of the placenta was perturbed or whatever. So they used it to see what, they had some slides, and they never really did anything with it. He says, but I have the slides. So let's take a look. So here is what it is, because we put it in a brief review that I was asked to participate in. And basically what he had is 17 weeks here. This is the human fetus. And this area here is going to become a limbic area. And as you go from here to C to D to E, it gets progressively differentiated. And here is a slice from each one of them, A all the way to D. And I don't think you have a lot of trouble seeing that the density is very different as you go from here to here. He also had some tissue from 20 weeks, and again, prospective limbic areas going all the way out like this. And at high magnification, this prospective limbic area, you see it here, these neurons, at least those of you who can see from close up, they start to look like pyramidal neurons already. But by the time you come down here, these are just little dots, these laminate areas. So it suggests that there is a difference as predicted that in development, in the timing of development, that gives us a difference in sight. And then we looked at the progenitor zone, and the progenitor zone is much simpler over these limbic areas than it is over here. And you see, we had to cut it out here because it was too long to put in this brief review that I was writing. So there are big differences in the progenitor zone as well that help explain what is going on. So the gray and white matter architecture in monkey correlates closely with the architecture in humans. And we can use these experiments that what we have learned from the monkey to, and then use the structure as a proxy to understand how many connections develop in the human. And so you see on the top is the human, and you see on the left there is the myelin, missile, and myelin is on the right, and below is the monkey. So we see parallels in what happens in monkeys and in humans. And so this was done by another very talented colleague, now a colleague in the greater group that we have, and he looked at the correlation between neuron density and axon density in monkeys and in humans. And you can see that they're not identical, but they're parallel. And so there are, this is Vasily Zikopoulos, and again, Miguel and myself. So the structural model has further explanatory power as the plasticity stability continuum of the cortex and the preferential vulnerability of some areas to psychiatric and neurologic diseases. And I would like to just give some examples of each. If we look at it, this again, Miguel's work. Limbic to laminate areas, some features here that vary. And this is the neurofilament protein SMI32 that is not as strongly expressed in the limbic areas, such as this one, but you can see a differentiation in this nicely laminated area. And the same thing happens with myelin. Limbic areas have very low myelin, and these laminate areas they show better myelin. So it's very interesting because these limbic areas are flexible. They're the ones that we associate with learning, memory, emotions, all of these processes that require mutability and flexibility. And it's not like the laminate are rigid, they are not, it's just all relative. So to give you now some examples about what may be happening in disease, I'll give you some examples in autism. And people had talked about autism. They made observations that the frontal cortex is enlarged in children with autism. So the first time I heard that, I asked them, it was a small conference. We were supposed to sit and think about autism. We had done nothing on it, but they wanted to speak with people who were doing different types of work. So I asked them, is it more, what is it about the white matter? Is it more axons? Is it more glia? Is it more myelin? They didn't know because they were counting pixels, and that's what was happening in imaging. And so they didn't know. I thought, well, maybe we should really do something to try to see if we can help them a bit with this question. And so it wasn't as easy as I thought because it's just even getting the brains. But to make the long story short, the superficial white matter in this area, the frontal lobe, the anterior cingulate is particularly in people with autism who had died and donated their brains, had more, they had more branching of axons, and they had more thin and medium-sized axons. And this is, again, Vasily Zikopoulos who did this. And the opposite was seen for thick axons. When people who talk about autism, they said a lot about the frontal lobe talking to itself. So the anterior cingulate, the thin axons are the ones that form synapses nearby. So if you really think about an axon traveling long distance, would be like my arm here and forearm. And when it gets to the point to connect, it's like my fingers, it splits into smaller axons. And he had a lot more of this. And so it was connecting, obviously, to nearby areas. And whereas the distant ones, axons that go from the cingulate to sensory areas, et cetera, were not, they were fewer. And so these were people in their 30s and 40s when they died. And so it's interesting because when you see people with autism, when they get, if they're high functioning, if they get interested in something, they focus on it and they get glued on it. And it's almost impossible to shift their attention to something else. And this is why it was thought that the frontal lobe is just talking to itself. And maybe this is why a lot of people who are very high functioning also are good problem solvers, because you really need to focus to solve a difficult mathematical problem. So we followed in that, and the axon growth protein, which is called GAP43, it was really elevated in axons of people with autism. This is a protein that is expressed everywhere during development, but in adulthood, it subsides in most areas, except in a few areas. And guess which areas have it? The limbic areas, and especially the anterior cingulate. So in these autistic cases that you see on the right, there was more of that, and it's shown in red, and it was inside axons that was shown it. And so Vasilis also found that when you look, there was more branching of axons in the superficial white matter, and you can see it here. And we also found something, because we were using some areas as controls. The anterior cingulate was a good place to start, because this is an area that is associated in humans and animals in attention and shifting of attention. And so it was there was a good place to start. But we looked at the orbitofrontal areas and some lateral areas. And in the, to our surprise, the orbitofrontal in the myelin was thinner. So we didn't really know, is this just something that is just another defect, or are the two of them related? And it's a little bit too complicated to explain, but basically we piece it together by looking at the normal development, seen in gray here, of the different areas that existed in the literature. And the ACC has a short period of migration. The orbitofrontal has one that is a little bit longer, and the lateral prefrontal areas even longer. And again, and sort of the expression of GAP43 and myelination also varies in different areas. So it turns out that GAP43 and myelin are mutually antagonistic. We didn't really understand this. And then I found a paper. I was very happy to see it, because that's what they saw, which means that if there is too much GAP43, axons keep on branching, but no myelination. So you first extend the axon and then you wrap it. And so if there is an interplay of these, it can help explain why in one area you get more branching, but the myelin is not affected. And in another, the axon branching is not affected, but myelin is affected. And why in some areas, like in the lateral prefrontal, none of these, at least these two parameters, were affected. So you can find these features that may be useful. And so in the course of development, in another study, the axons which connect nearby areas increase significantly in individuals with autism, and in controls, they remain about the same. And the opposite is true for thick axons. So we can really see that as it develops, it goes in development. So limbic areas are plastic and engage in learning, memory, and emotions, but are also vulnerable to disruption and disease. Diseases that affect predominantly limbic areas may perturb processing of context, because they innervate the upper layers, layer 1, which gets the apical dendrites of the pyramidal neurons from below. So it may be some way of actually providing information that can pull a lot of information together and provide context to interpret what is happening, the environment, the social interactions, and so forth. So diseases that have their role in development show variable expression in adulthood. So people have asked, why is it if something happens in development, why is it that you have such a large variation in expression in autism. Some of them have, these people have no language and lots of seizures, etc. Some of them are, quite normal, and some of them are on the genius range. Well, if you go back to development, one way to help explain and investigate in the future is that it depends when the perturbation occurs, whether it's a combination of genes that affect development or whether it's some perturbation in gestation that happen at different points. It's going to also affect the development of the neurons and that are going on at that time. So it will be variable. And so I think that it at least tells us why we have such a range. And it's not only that, it be also schizophrenia and the other diseases as well. So what are some other implications of the systematic variation? And one of them is inhibitory control. And so the frontal lobe is associated with this function. People with even mild cognitive decline showed that they had loss of inhibitory control. Bob Knight and his colleagues saw that in patients. And so what was really happening with them, but we really didn't know how inhibition is exercising at the level of neural circuits. They saw it in patients. You see sometimes people who have even mild cognitive decline, such as somebody in an advanced stage, and they avoid noisy environments because they cannot follow conversations. And so what really helps you drown out the noise when you're in a situation like this, and there's a lot going on around you, but you're able to figure out what somebody is talking when they're talking with you. So we looked at it at a different level. So we can now study the pathways, and here's at the confocal level is another level, and the pathways that we can label in animals. And you can see the axons in green and the inhibitory neurons in red, and we can see close appositions and then go to the EM, as Javier told us so beautifully earlier. So we can do that. And the interesting thing about primates is there are three largely non-overlapping neurochemical classes of inhibitory neurons labeled for calretinin, calbindin, and parvalbumin, and they are found in different layers. It's quasis. The nervous system is really more wishy-washy than that. But as you can see there, the calretinin neurons on the right, they are in layer 1 and 2 primarily, fewer in other layers. And they innervate other inhibitory neurons, and they are known as disinhibitory. And the calbindin neurons innervate more medial to distal parts of the dendrite, and the parvalbumin they have very strong perisomatic inhibition of neurons. And so we can put all of this together. And so let me just give you a couple of examples so that we have some time for questions. As I told you earlier, the phylogenetically old limbic cortices behave as global feedback systems. And one of them is in the anterior cingulate, area 25. And you can see it here. This is the work of one of my graduate students, Mary Kay Joyce, who's now a postdoctoral fellow. And what she looked at it as the whole connectome of area 25, and saw that it really did behave as a feedback system for the most part. And in subsequent, and looked at some of the implications of this. The lateral prefrontal cortex is thought to control activity in other areas. And for example, this area 25 is hyperactive in depression. And this is one of the most consistent findings that they have seen in humans, human psychiatric diseases. But area 25 has few direct connections with the lateral prefrontal area. So how does this inhibition occur if it's going to really control it to some extent? And what she found was that there are parallel pathways, serial pathways from lateral prefrontal areas that reach area 25 through the anterior cingulate, area 32. And so you can use traces in a clever way to show which neurons project here and how these neurons project down here. And you can see that over there. So she studied this all the way from the system to the synapse. And I wish I had more to show you in here. But this is a reconstruction in 3 dimensions. This is the little connectome of this area. And so the blue shows the terminations in this area here, in area 25 from 32. And the green is the dendrites. These are the dendrites of excitatory neurons. And in red, you see the inhibitory neurons, just like the traffic lights. No, I can't give this example in Boston because they don't observe those rules as they do in most places. So, so this is a very small part of the of area 25, and is studied with synapses from this region. So there is a way to look at it. And so some of the implications for that is that there is dysregulation of serial paths from prefrontal cortex to 25 in depression. And the question is does this occur? And one of the things that we found in 25 is there is an abundance of calretinine. These are the disinhibitory neurons, and they're found in the upper layers. And other people found them also in humans. Zilli, Simons, and Calomero Gallagher. So they found the same thing happening in humans. Palomero Gallagher, I'm sorry. Yes. So, and there is a new therapy for depression, ketamine, and ketamine seems to work very fast. And it's an NMDA receptor antagonist. And so you can see in this area here that we labeled the calretinine neurons with an antibody, and NR1, which is the obligatory subunit of an NMDA receptors, and you can see that there are a lot of them, and the excitatory neurons also have a lot of these receptors. So ketamine may block them, and because these are disinhibitory, it may actually block local excitation within the column. And we don't know if this is what is happening, but it's a potential mechanism. So if we now superimpose the structural model with the system to the systematic variation, as I told you before, on the distinct types of inhibitory neurons, we can start to make some hypotheses about what happens. Because when you look at this distribution of the different types of inhibitory neurons, they're very much the same in areas, but their relative density is different. So in some areas, the limbic areas, for example, don't have a lot of these parvalbumin inhibitory neurons that are very powerful. But you need to do this analysis in a lot of different areas because it's not a global kind of things. The lamina distribution seems to be like that, but there are some exceptions there as well. So when we think about signals from the external environment, we know that they get propagated, they hit your retina or your ears, and it goes from there to the cortex and to high order association areas beyond. But what about other functions? Most of what we do is not really interpretation of the sensory environment. You can do a lot of things with your eyes closed, thinking about it. And so how does that come about? We know that these limbic areas get a very, very strong input from the amygdala and the hypothalamus. They also get unilateral input from the hippocampus. And via feedback pathways go to these lateral areas. So it's just the opposite of what we see in the sensory area. So if you initiate the activity in the motor system, which is really a continuation of what I'm talking here, it shows the opposite sequence than the sensory areas. And I think that I wanted to give you an example because we are very sensory centric in the interpretation of the brain. So I wanted to close with what the implications might be for evolution. And I showed you earlier that this figure here and that these areas develop earlier and these areas become more and more differentiated. And if that, it culminates into this occipital region, which looks really very nice over here. Are these the newcomers? We think so. That's where it pushes us to think. It's controversial, but I don't think it need be based on the structural issues that I talk with you about. And so cortical systematic variation provides the framework to understand the organization of connections and to view the cortex as a continuum of areas with relative stability and plasticity, and disruption of nodes in disease based on their relative plasticity or stability, and the relationship of connections with its inhibitory system, and the sky's the limit for studying systems like that. The ability of some areas to control others in disease so that you don't have runaway activity. And the orderly maps and connections may allow flexible recruitment of areas through the cortex, thalamus, and subcortical structures such as the amygdala, hippocampus, etc. in behavior. So basically, what this type of analysis shows is that you can, it's almost like having the railroads that are laid down there. It doesn't tell you about the trains coming onto those railroads, but it tells you that they are organized in such a way so that it can, that flexibility and stability are maximized for processing information, just something to think about. And this systematic variation of the cortex is laid out in development, and now we have a little bit of support for that, and we need more, and may also reflect the evolution of the cortex. And I talked about some of these people who helped in all of these areas in our funding agencies, and thank you for your attention.